Hi everyone, I'm Francesca Cuturello and I will talk about machine learning approaches in structural biology, so very different perspective on the topic, let's say, but hopefully, well, first of all, I want to thank for organizing these meetings, which I think they're really, they could be really useful to find a common ground on which we can hopefully cooperate in the context of this project. And I want to start by presenting my group. So I'm part of the RIT institution at Area Science Park, whose director is Stefano Cozzini. And inside this institution, there is the Laboratory of Data Engineering, which I'm part of, and which is a highly multidisciplinary group, let's say, because it has, and it's very young. It was born basically not even 2 years ago, and it's growing and growing. And yeah, our backgrounds are very different. They go from physics to mathematics to informatics and genomics. And there are many different skills around, like machine and deep learning or high performance computing. And the combination of these skills and our curiosity and interest basically led us to study machine learning and structural biology. So what I want to say in advance is to apologize for, I want to show you pretty much what we did in the course of this couple of years. So I will go very, very fast and I will not be able to explain in detail the techniques, but hopefully if you're interested, we can for sure talk about it more deeply. And just a little bit of history on computational approaches for molecular structure prediction. Well, of course, there is molecular dynamics, which consists in sampling conformations trajectories by minimizing a potential energy. So here the main effort is in tuning the force field parameters in order to have a reliable prediction out of the dynamics. And then there are covariance models that they basically exploit sequence information only. They're really fascinating to me, and I was studying these models during my PhD at CISA, actually. So these models rely on the concept of homology. So when we have a protein family, so a set of sequences that are related by because they mutated from a common ancestor, and they still preserve the structure while mutating, then we can align them in a multiple sequence alignment. And when we look at two residues positions in a sequence of this multiple sequence alignment, that is involved in a physical interaction in the structure, then there is a simple idea that the two residues should undergo coherent mutations, compensatory mutation. So this is like a signal called covariation or coevolution, and it can really be exploited to find some information about the structure. So when speaking about models that rely on sequence information only, we come to speak about machine learning methods, because in recent years there was a great advance brought to the field by these techniques. And for us, when we started, basically it all started by when we had the opportunity of studying this DPCFAM dataset. This is a dataset produced by the DPCFAM algorithm that is that was developed in CISA in the group of Alessandro Laio, with some of the developers that are currently working on our group. So that's why we had this chance of analyzing this database. This is basically an unsupervised density-based clustering of the sequence space, and in particular of local pairwise sequence alignments. So in the end of this pipeline, which is quite simple in the end, we obtain domains of evolutionary related sequences, like in the PFAM style. I think you all pretty much know PFAM as a standard let's say, the gold standard for homology detection and sequences classification, we don't have a ground truth here. So what we did, we compare with the PFAM database, basically. So see if this classification was consistent, and we found it was when clustering the UNIREF 50 database. And also, this method was able to detect some domains that were not that have not been annotated before, and these were actually analyzed by the PFAM team and included in the PFAM. So from this point of view, it was really a success. And just recently, we applied it to the human gut proteomes dataset, and we find really consistent results in terms of distributions. So showing that this approach can be used also on metagenomics data, which is not for granted, let's say. And well, I forgot to say, I won't mention all the single person involved in the project because we're trying to do it. So an ongoing project is the DPC tract. So when we want to take a very similar approach, so the density peak clustering developed at CISA to cluster pairwise, local pairwise structural alignments now. So exploiting two things. One is a relatively recent algorithm, which is the FOLSIC for structure alignments, and the other one exploiting large database of structures. So in this way, we aim at refining protein families classifications, and maybe explore and investigate the hardest cases, which are in the twilight zone. So when the sequence similarity is really low, gets really low, but you can still have structure that could be quite similar to each other. And what I'm kind of hiding here, but it's just the link to the next slide, is like, why do we have suddenly all this large database of structures available thanks to the release of the AlphaFold database, which relies on the AlphaFold 2 algorithm that was winning, I think, CAS13 competition. And so I guess everybody in this room at least heard of it, if they didn't use it already. So yeah, this is a model that takes in input a protein sequence, and it will give you in output atomic positions in the folded three-dimensional structure, so basically a PDB. And I want to underline already something. First of all, it seems like it takes an input a single sequence. Actually, the first thing it does is to build multiple sequence alignment. So it looks for the protein homologs. Then there is a more recent version that was trained on single sequences, but it gives much worse results. So you'll see why this is important in what I will say in a bit. So when we look, this is a very complex model. It's also modular. It's made of many blocks. And when we look at the first block, so the evoformer block, we notice that it fully relies on a relatively novel architecture, which is the transformer architecture. Is a deep learning algorithm that is very much studied recently. It's also behind ChatGPT, just to say that it has many important applications also in other fields. And in fact, a little bit of history of the transformer and just a vague explanation of what it is. So it was, it came from the context of language processing. So the idea is that you pass a sequence of tokens to the network, where the sequence, it can be in the context of text sentence, and the tokens are the single words in the sentence. And we can we can imagine to mask some of these words and ask the network to predict the probability of the missed word. So basically, how is it trained? Is it trained by this best masked likelihood, in which basically one wants to maximize the probability of observing the correct token at a masked position given the unmasked token. That's why they are called context-based, because they learn from the other tokens around. And yeah, that's also called the self-attention mechanism. So from here, we can see how it can be quite straightforwardly applied to the protein universe, in the sense that now the sequence of tokens can be sequence of amino acids that can be processed and masked, and a fraction of them that can be masked to read the training and ask the network to predict the identity of the masked amino acids. And so really these networks are able to capture higher the correlation between the token in the sentence, in the sequence in this case. And that's why this architecture is really much suited to large pre-trainings, where from large pre-trainings we mean that we can train it on a large corpus of text, for example, or on the UniRef 100, so basically all the sequenced proteins, and then use the learned representations that are hidden here to fine-tune the models on specific downstream tasks. And yeah, they've been found very powerful for this, and I wanted to convince you also because they're not used in the protein context only from after fine-tuning on a specific task, but they are informative already at the pre-training level, because in the hidden layers, the contact maps are actually learned and encoded, and we can extrapolate just from the pre-training the contact map of the proteins. So when we come to contact maps, we have to mention an upgrade of the model that was adjusted to take as input a multiple sequence alignment and no longer just one single sequence, because this was found to improve the contact prediction. And we were really interested in this model at the beginning, and so we started studying the impact of the different multiple sequence alignment in many sense. So we looked at the level of similarity of the sequences in the alignments. We looked at the different strategies to detect homology. So basically we compared PFAM with the TPC-FAM strategy, and respectively because there are some technicalities here, we parsed 900 sequence families, in which for the MSA transformer model we need a PDB inside to check whether we are to check our contacts, basically the predicted contacts. And so this is this constrains a bit the data set. So we first compared PFAM with an algorithm that is an automated algorithm to search for homologous sequences in large database. And actually, this is MSec2, which is used by AlphaFold to construct this multiple sequence alignment that I talked about. That actually is the input then of the AlphaFold model. And so it was interesting to look at. And also we compared the PCFAM with the HH-Blitz search, which is, let's say, it has the same idea of MMSEX behind. So to search for homologs in large databases. And this is the particular trait that the MSA transformer network was trained on this type of alignments. And what we see, the main results here is that when constructing these families from representative proteomes that are more filtered by similarity, so the sequences that are inside are more different, the models predict better the contacts. This is the main take-home message. And this is a sign of the fact that actually covariation and coevolution is still used and important to learn the contacts here. Then we did some cool things like continue the training, the MSA transformer training on the TPC-FAM dataset, because we saw from some results from some experiments that the MSA transformers was not quite able to generalize on the different homology detection methods and the different multiple sequence alignment construction strategies. And actually, the continued training was beneficial for the accuracy of contact prediction. Then we used AlphaFold. And so for AlphaFold, we needed to select 60. We want to compare PFAM, DPCFAM, and the default MMSEC search performed by AlphaFold if we just pass one sequence to it. And this was interesting. We selected 60 families that had the same classified portion of sequences so that they were comparable between PFAM and DPCFAM. And we were particularly careful that they shouldn't contain PDBs here, because we didn't want AlphaFold to use this information in any way. And we see how actually the internal accuracy of the AlphaFold model is better when using custom dataset, custom multiple sequence alignment with respect to the default search. So all the because AlphaFold has, I think this is one of the most powerful traits of AlphaFold. There is an internal measure, there is the PLDDT, there is a train itself.
An AlphaFold parameters. So
Yeah.
to have an external check.
We have the PDBs here, so for sure we can do this check. I didn't do it yet because honestly, I don't think here, these are really good alignments. You can see it already from the MSA transformer contact maps. And yeah, and I'm not sure that it will change so much the structure prediction in the end at this level, let's say. But yeah, for sure we can check because these were, let's say, a subset of the 900 families. So we have the PDB here. It's just we're not using it for this experiment because we don't want to bias. But yeah, it's a good suggestion, actually. We should look at it. Maybe we look at the root mean square deviation between the PDB and yeah, thanks. So, but the take-home message here is covariation is important. This coevolution effect should be important. This makes it even more interesting to study a very important subset of protein language models. We rely on single sequence because it looks like kind of magic how they can learn something out of it. And so that's what we did. We did a more, we studied this model really in depth, looking at the hidden layers of the model. So all machine learning methods project independently of the kind of operation they perform internally, they project the data in a really high dimensional space. And this is the representation space. These are really vectors, if you want, but really in high dimension. And what we looked at is, first of all, is basically the geometry of this representation space from a global point of view. So we were estimating the intrinsic dimension in the layers of the network. And intrinsic dimension, you can see it as the dimension of the smallest space in which you can project your data without loss of information, basically. So when we looked at this in the layers, which is the x-axis in the plot here, we see a three-phase behavior. And the interesting thing here is the universal trait of this behavior, which is shared across many different models. I just show ESM2 here, which is the most recent and accurate among the single protein language models, but this is shared also across the older models, and also for different sizes. So the models can have different number of parameters, and still they have this three-phase behavior. So we have an expansion of the intrinsic dimension from the input dimensionality, and then it reaches a peak, and then we have a contraction of this dimension to a plateau. So many layers which are capable of doing this really low dimensional mapping of the data. And then at the end, we can see a final ascent that brings back the intrinsic dimension to the one of the input. And this, we think, it's quite clear that it's because the network is solving the reconstruction task that I was showing before, the BERT task, the construction task. So it needs to go back to the dimension of the input. Well, all this can be studied also locally, and maybe that's more like it's easier to interpret when we look at things locally. So what we looked at, the nearest neighbors overlapped. So we take a representation in a layer. We looked at the first k nearest neighbors in the representation space. So imagine these are vectors, so you can compute distances. And we look at how the neighborhood is preserved from one layer to the other. So at the beginning, so the orange curve. So at the beginning, these representations are all incoherent. The neighborhood changes from one layer to the other. Then you start to see coherence. So basically at the plateau, where to the layers that correspond to plateau in the intrinsic dimension behavior, we see that basically the representation keep the same neighborhood in the layers. And another things that we notice is that if we compare with the scope annotated dataset, so we looked at the identity of the neighbors. So we want to check if, for example, a neighbor of a given sequence representation is part of the same superfamily. We find it is, and we find it with a 6% improvement for free with respect to doing it at the last hidden layer, because people in neural networks always use when they want to use this rich and informative representation, use the last hidden layer, because they say this is where the network has learned the most. But in this case, we are kind of showing that you should take plateau layers if you want to be general. And actually, we see this improvement in superfamily members classification when we use the plateau layer. So this was all to give you, to deliver the message that these models, sequence representations, and do encode biological properties. And the community now is really pushing some of the interesting implications that this could have, or applications, and also the challenging aspects, for example, in the context of mutations, to predict the dynamics of evolution, to infer the fitness landscape, predict the effect of point mutations, which AlphaFold is still not really reliable on doing. Then there is the context of interactions, in which people are studying protein-ligand interaction, protein-protein interaction, still with these approaches. And then there is the problem of conformations. So I want to study, basically, here I will just give you an idea of the first two, so mutations and interactions, through some, because we're really working on these topics now. So I will do it through some works that we're doing from other people, but they are very inspiring for us. And on the conformations, I won't have time to do it, but I can just say that we started tackling this problem with Alberto Cassetta on the dystrophic protein, and on this flexibility problem that he was talking about, because there are very promising techniques in this context also. So, but we are really interested about mutations right now, mainly. And here I want to give you an example of a recent work that to me is really interesting, and it showed us that we can really do something in this context. So what these people did, they took a single sequence protein language model, and of course, they took the last hidden layer representation, as I was saying. So and it's called in this notation X of A, where X of A is the sequence. And then they said we can just define the evolutionary velocity from mutating from sequence A to sequence B as the difference in the BERT likelihood that I was showing before. So the probability of observing a given amino acid given the context, while the probability of observing the other amino acids in sequence B given the context. And the thing here is that this calculation is performed on the M subset, for which after aligning the two sequences, imagine that you have a subset M of residues that are discordant. So they are mutated, one with respect to the others. And you can define this evolutionary velocity in this way. Then they constructed a directed graph where each sequence representation is connected to the first k nearest neighbors, still in representation space. And they define the transition matrix in a very natural way from the evolutionary velocity they were introducing before. And at this point, we have basically a directional graph where this is a diffusion process actually, and it needs to be solved to find the roots of this process, and these are the fixed root of the diffusion process. Once you have the roots of the process, you have an order in pseudotime, and you can identify mutations and also the order in which they appear. And I'll jump to the results because I think they're interesting. They are actually, also I chose this paper because it's quite visual. So then we have a vector field, a velocity vector field, and it's in high dimension because we were considering in this directed graph more than one neighbor, no? So we can project it to the dimension through a UMAP dimensionality reduction. This is just needed for visualization. And we keep the arrows to represent the vector field. So when we color by the actual, on this data set, it's a nucleoprotein of influenza A virus, that it's a really good test case for them because it has a densely sampled sequence evolution, and it has a relatively low mutation rate because it mutates, I think, on average, one amino acid per year. And here, basically, the natural selection is shown in the form of host immune pressure. So when we color by the actual time in which this mutation appeared, we see that the arrows go in the correct directions, let's say, with the actual time ordering. So there is a correlation between the flow velocity and the known sampling time. And then when we go to the plot, the next plot, we see the roots of the processes, which are colored in black. And interestingly, they defined four roots, unsupervisedly, let's say. And then they see that these four roots actually correspond to actual beginnings of four different pandemic events. So this is nice. And then again, they looked at subtypes of this nucleoprotein, and they see that these are well grouped by this velocity flow. And also there is one detail that they claim it's nice because there is this red subtype here, which was predicted to have originated from the blue subtypes around in the phylogenetic from a phylogenetic analysis, but they predict it to be independent origin, and actually this is confirmed by recent epidemiological data. So let's say this is a more robust approach with respect to phylogenetic analysis. And yeah, they very quickly here, they also showed it for the spike protein, which of course it's much more challenging with respect to the other dataset because the time scale in which the sampling was performed much smaller. And so, yeah, but still when we plot the trajectory in two dimensions, we see by looking at the actual time that this is kind of well grouped by the clustering. And this is, let's say, less visible with respect to the other case. And so that's why they chose to show two particular mutations which are interesting and that were actually classified to be in the correct time ordering by the algorithm pseudotime when compared with the true time in which this mutation appeared. And so, yeah, from our side, we are really studying this kind of problems, and we can do many things here. We can use, for example, our results from previous works that we did in protein language models. For example, in this case study that I just shown, they were using the last hidden layer representation. Maybe they could get much better result by using other layers. And then we have been working already, and we are still working on developing some generative model to produce high-fitness sequences. And we started doing so with a preliminary work that it was more, let's say, theoretical from some point of view. So using a really advanced machine learning techniques, that is adversarial attacks on these protein language models. And we have a preprint on that, but we are really pushing this further. And then, of course, we lack the interplay with experimental approaches, which would be really, really beneficial for us to learn and also to see where we can help. And yeah, just if I have time, there is a really quick case study, but here I just wanted to show you how you can use AlphaFold to do something that AlphaFold is still not able to do, let's say, so pushing AlphaFold to the limits. So basically, this the David Baker group was trying to predict protein peptide interaction with this model. In the end, in this work, they're just able to say if one peptide is bound or not bound. So they're not giving a notion of affinity in any way. But still, it's something, I think it's nice work, and we are also interested in this kind of approaches. So basically, what they did very quickly, they here, I mean, AlphaFold is not yet suited for passing as input a complex. So the main effort was to work on the input template to be able to do this. And they do it with actual quite simple tricks. And then the main part is you run AlphaFold, you run your structure prediction, and another internal accuracy of AlphaFold, which is somehow complementary to the PLDDT that we were mentioning, is the predicted aligned error. So they looked at the predicted aligned error between the peptide and the major histocompatibility protein, and they see, and they put basically a classifier on top of this. So they fit the predicted aligned error with a logistic regression, which uses the label bound, not bound, from a large data set of available labels. And then they go back to AlphaFold after doing this. They fine-tune the AlphaFold model, and then back again to the structure prediction. And then they do predict the structure prediction accuracy, and back to minimize the structure prediction accuracy. So it's a back and forth to refine the AlphaFold parameters to actually be able to tell you whether a peptide can bind or not. And yeah, I mean, computationally right now, maybe only really you have to have great resources, but we do, so we are also thinking about these kind of problems. Yeah, here that's so basically this is a fine tuning by minimizing a hybrid loss. And they showed that they reach the state of the art from the computational side. I mean, this is still the pink line is the state of the art, which is NetMHC-PAN, which is still a neural network, a machine learning model. With respect to the AlphaFold alone, let's say, without the fine-tuning with the logistic regression labeled bound, not bound. And they see that they actually make it to the state of the art, and also they claim they are more robust of the net and MHC-PAN. I don't show, I just say it with a sentence, but they do some experiment to show that basically their approach can be extended to not only 9 MERS as peptides, but they can it can be suited for 10 MERS, 11 MERS, I don't know how to call it, for longer peptides, while the other approach is not, because it has been trained on 9 MERS. So this, they claim is more robust. And with this, I missed a conclusion slide, but I think this is just an idea of what we've been doing and where it led us, basically, to study. And from our perspective, we have a lot of things to share with experimental structural biologists as well. So